Hey guys, how's it going? My name is Joel. Hope everyone's doing all right. I'm going to be giving a presentation on this paper from 2019 done by Karen Liu at King's College London. Its title is Induction of the Neural Crest Stem Cells from Bardet-Biedl Syndrome Patient-Derived Human-Induced Pluripotent Stem Cells. The main topic of this research is developmental genetics and the studying of the development of this complex set of cells named neural crest stem cells. To get any idea why this topic might be important, there are two, we could say, themes that I need to explain. The first one will be neurulation or neural induction, and that's modeled here on the diagram furthest to the left. This is neurulation, this is formation of the neural crest, and you can see on the bottom part of the diagram this structure where you see green cells. Those are the neural crest cells, and they're going to be migrating, they're going to be delaminating and then migrating out of the neural tube to become all sorts of tissues, and those are outlined here in the diagram furthest to the right. So you have dental tissues, you have bone, cartilage, smooth muscle. So there are many of these involved. So you can start to get an idea as to why the dysregulation in the development of this structure can be highly consequential. And indeed, the diseases that occur due to the poor development of the neural crest are known as neurocrestopathies. Second are ciliopathies. These are diseases that are categorized by the loss of function at the cilium. The cilium, although having many roles, it is particularly relevant in this case for development. We know that the primary cilium has an active role in neural development, and we know that the cilium can be linked to many craniofacial anomalies, such as neurocrestopathies. Shown here is a diagram of the cleft on the bottom, and on the far left is a diagram taken out of the paper that highlights how BBS proteins, these are Bardet-Biedl syndrome proteins encoded by BBS genes, are actively involved in signaling at the cilium. Although the link between this signaling at the cilium and that of neural crest migration is not yet understood. So Bardet-Biedl syndrome then becomes a suitable model to study the migration of the neural crest. It's a ciliopathy. Its phenotype resembles that of neurocrestopathies, including craniofacial abnormalities, and it's easy enough to obtain samples from patients, and the genetics are relatively known. In this case, the researchers have focused on the BBS10 gene as it is the most common mutation found in BBS patients. So then let's take a look at what the concept of this research was. So here's a really quick summary of what it is that we're going to be looking at. It's an example of organoid disease modeling. It is the first to show neural crest induction being modeled in human-induced pluripotent stem cells, and it is the first paper to really want to find out this role of the cilia during neural crest development. So without wasting any more time, let's just get into how they made these organoids. So different cell lines are established using different mutations. One is the wild type over here. Colg and Kegd are the two wild type cell lines that have been derived. And then there are Zyri and Lag. Zyri is shown in purple. The top bar represents one of the alleles with a non-synonymous mutation. Then below it, a different allele with a nonsense mutation. Directly below it is Lag in orange-brown. It has a synonymous mutation on one of the alleles and a truncation on the other. More on how these organoids were established later, but right now, just looking at this first figure from the paper, we can see on the left how the pluripotency marker OCT34 was checked for fluorescence in these cells. So the first line is of the wild type, and we can see it is highly staining OCT34, and there is no significant difference in the staining in both Zyri and Lag. This indicates that pluripotency has been achieved with equal significance in all of the cell lines. However, if we look at primary cilia, the gene that we're looking for over here is ARL13B. The difference may not be so apparent, but there is a slight significant increase in primary cilia in Lag, but not in Zyri. So what this might suggest is that BBS10 may not be needed for formation of primary cilia, at least at the pluripotent stage. It is important to stress the lack of significant difference in OCT34 expression in all of these lines, showing that indeed all of the organoids are fully pluripotent. So this slide is very important for understanding the entirety of the paper altogether, so it's worth spending some real time understanding it. We can tell over here that there are 3 moments in the induction of the neural crest that have been specified by the researchers. This is at 0, where we say this is the stage of pluripotency where we expect to see such genes as OCT34 and NANOG being expressed, and then At 48 hours, we expect to see neural plate border identity, that's PAX3, PAX7. And then at the neural crest identity at 120 hours or 5 days, we expect to see the expression of such genes such as FOXT3, SNAIL2, and SOX10. The way that we induce the neural tissue is done via Wnt signaling, but by GSK3 inhibition. It is a protocol that was established by this paper at the bottom, Lung et al. The protocol is fairly simple to understand. It is subjection to neural crest inducing media, which includes GSK3 inhibitors, over a period of 5 days. So here are some photos taken at different time intervals during those 5 days of induction. And as you can see in the beginning at 0 in that pluripotency stage, as we go down the column, um, you can see that there isn't much of a difference between the cells. As we said, they are all expressing the same pluripotency markers with no significant difference. So what you can see is an equal amount of isolating in these islands, and there is some protrusion around the periphery of these. But it's only until we start seeing as the time goes on that at 48 hours, marked here with a blue arrow, the wild type cells have begun to delaminate out of these structures and they're moving away. They are starting to migrate, and that's just not seen in either of the mutant lines. In particular, leg is extremely conserved. At 96 hours, finally, Zyri and leg start to show these delamination features, these cells moving out of those colonies, but they do so at a delayed rate. And you can see that post 5 days, what we can see is that at confluency there is a much lower density of cells in both Zyri and leg in comparison to the wild type. What these data are showing us over here is that although some neural crest identity is seen, there is some delamination, it appears to be delayed in mutant lines. And this is the same data, but zooming in a bit further and looking at just Zyri and the control, looking at just at those 96-hour results, you can definitely see that the density of cells in the mutant line is far less. And you can see that actually the wild type cells have started to migrate at 72 hours rather than at 96. So the researchers now wanted to see what were the genes being expressed at these different stages. And for that, mRNA was isolated at 0, 48, and 120 hours. Importantly, what the researchers expected to see was a gradual decrease in pluripotency markers OCT34, NANOG, and CMYC as time goes on, and an increase in the neural plate border identity genes and neural crest identity. And we would definitely expect that these genes would be at their highest expression between 48 and 120 hours. So this is expression of the pluripotency genes, and the wild type lines are the ones in blue. And we can see that there is a quite elegant decrease between, iPSCs to 5 days. That is seen in all of them. Although, however, it is worth mentioning that Zyri has decreased the least since its pluripotency stage, and this can be seen in NANOG, where at 48 hours, where the other two are already at 0, Zyri is still expressing quite high levels of NANOG. And this is suspicious because it could indicate that because there is expression of NANOG, a pluripotency marker, maybe these cells are not responding to other cues. This theory is further substantiated when looking at neural plate border genes. Looking at just 120 hours on the far left, PAX3 is significantly upregulated in the wild type in comparison to any of the mutants. This is also seen in TFAB2 between the wild type and leg, and it is also seen in PAX7 between wild type and Zyri. Unfortunately, data was not shown for leg due to technical difficulties. So the researchers are now thinking that the delay that we saw in previous slides was due to the inability of the mutant lines to recapitulate the same level of signals as in the wild type. Looking here at neural crest genes that we expect to see after 5 days, it is interesting to see that in FOXT3 on the far left, there is a significantly increase in expression of FOXT3 in leg iPS cells, which is not what should be happening, as you can see by the wild type. And focusing at just the 5 days across all three of them, the same pattern emerges. There is high expression of these genes in the wild type, and there is poor expression of these genes in both of the mutants. SOX10 here is an interesting gene because it is actually required for cells to migrate. And you can see over here that these cells do have an inability to migrate because they are in fact downregulating SOX10 at 5 days. These are also quite interesting to see. Brachyurin, the first one on the top left, is a marker for mesoderm, so we really wouldn't expect to see it at all as the wild type is showing. However, Zyre appears to be upregulating brachyury at 48 and 120 hours, which gives some more evidence to the thought that these mutants are responding to different cues. SOX2 is a marker of early pluripotency and also neural tissue, so we expect to see some of it, and that is indeed seen in the wild type. However, at 120 hours, we can clearly see that Zyre is down regulating this gene. ZIC1 and SIX1 are all needed for preplacoidal ectoderm induction, and these are also downregulated in the mutant cell lines. As most will know, there are key signaling pathways that are involved in all of neural development, and the main ones worth highlighting are Wnt signaling, BMP signaling, and also hedgehog signaling. Respectively, here is shown axin2, which responds to Wnt signaling, MSX1 that responds to BMP, and PTC1, which is patched1, which is a receptor for hedgehog. And what we can see over here is actually very significant. So axin2 and MSX1 clearly reveal that the mutant cell lines are not responding accurately to the signals that we would expect, especially Wnt signaling shown by axin2. And patched1 is for hedgehog, as I said. The role of hedgehog signaling in all of this is not really determined, but it does show a significant upregulation um, of a hedgehog signaling in Zyre and at least a pluripotency stage. So this again is more evidence that there are some signaling cues or some pathways being activated that are not expected. So some concluding remarks as to why this research was relevant. So, granted, not everyone's going to be interested in development or the genetics behind it as I might be, but there are some key things to learn from here. And one of them is that this is a proof of concept that this tool for modeling disease in a dish can be quite powerful. If we think about it, these cells were taken from their patients. And so it provides a personalized dissection tool to determine the cellular and molecular pathology of disease. These diseases are often complex and they have many genes interplaying, and these can vary from patient to patient. So, a tool like this allows not only diagnosis but prognosis and even maybe personalized medicine for these patients. Also, on the concept of studying gene function interaction, this is often really difficult in some animal models where double knockouts can result in embryonic lethality, even though we know that different gene combinations can actually exist in humans. So knock-ins become a thing that can be done in these organoids. And also BBS10 is a ciliopathy gene, and we can see here that the cilium does play a pivotal role, even though that might not be totally understood. This research does demonstrate that ciliopathic human-induced pluripotent stem cells have a diminished ability to respond to the signaling cues of GSK3 inhibition, and that was demonstrated quite well in the last slide looking at Wnt and BMP signaling after those 5 days of induction. The potential role for the cilia in patterning of the neural plate border and neural crest induction is still not understood. However, it can be further studied using models such as these. Lastly, I'd just like to mention that this technology, this tool over here, is highly adaptable, and we could be looking at different lineages such as bone, cartilage, and adipocytes and neurons. So the potential of these tools in other applications and for, like I said, regenerative medicine or dissection of molecular pathology in so many different diseases is possible. So that's all from me. That's the end of my talk. I hope you found it interesting, and I hope you have a rest of a great day. Okay, see ya.